AS Level Biology 9700, Paper 2, Variant 1, May, June 2025. Today we are going to discuss question number 1 and 2. And question number 1 is amylose and the triglyceride styrene are macromolecules. So here you can see explain why amylose and styrene are macromolecules, but only amylose is a polymer. Macromolecules are the larger molecules which are made up of many micromolecules. So here in this case, the amylose and styrene both are the macromolecules. They are the larger molecules, but styrene is not a polymer. Amylose is a polymer. Amylose, is a polysaccharide, which is a type of carbohydrate, and the amylose is the structure found in starch. So amylose is a micromolecule, and it is a polymer because it is made from the repeated linking of a single monomer, alpha glucose. Alpha glucose. So alpha glucose and will join with other alpha glucose and so on. So in this way, it is made up of repeated linking of a single monomer. So that is the reason it is called as a polymer. I would like to write in a short way, that is the because it is made up of repeated units of monomers such as alpha glucose. So this is the one point, and the second point is about the styrene, which is a triglyceride. Styrene is a triglyceride, and it is a type of lipid. Amylose is carbohydrate, whereas styrene is a lipid. So it is formed by different types of smaller molecules, one molecule of glycerol and other molecules of stearic acid. So that is the reason it don't have the repeating monomer units. No repeating monomer units. So it is not a polymer. Both of they are macromolecules, larger molecules. But amylose, carbohydrate, and styrene is a lipid molecule. So in this way, the amylose is made up of the repeating units of monomers, whereas the lipid is not made up of just like this. It is made up of glycerol and stearic acid. So in this way, it is not a polymer. Moving towards the next question B, and next question B is students use the enzymes maltase. Students use the enzyme maltase extracted from the fungus Asparagus or Oryzia to investigate the properties of the enzyme. It's a diagram of maltose molecule. So here you can see the maltose molecule is there. Now maltose, to break the maltase is an enzyme. So this enzyme will hydrolyze the maltose into two glucose molecules. And that process is called as hydrolysis. As if I would like to say there are two types of reactions. One, condensation reactions. Which takes place by the removal of water molecules. So the formation of glucose molecules, two glucose molecules will synthesize maltose. So when maltose will be hydrolyzed, maltose can be hydrolyzed by adding water. It will break down into 2 alpha glucose molecules. So we will write in this way plus H2O, and there will be the formation of two. It will break down into two molecules. H2 orange, and then it will be orange, H, 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 and then H, H, and H. So it will be C6, H12O6, and the same two molecules of alpha glucose CH2OH, H, OH, H. It means this, it will break down. This bond will break down with the help of this enzyme, which is called as maltase enzyme. So it will be broken down. It will be broken down into two alpha glucose molecules. And you know the alpha glucose molecules I have it will be broken down into 2 alpha glucose molecules. Little bit. So the next question is complete figure 1.1 show the reaction catalyzed by maltase. So I have made this. Water will be added into the maltose, so it will be hydrolyzed into 2 alpha glucose molecules. So state the type of covalent bond that is broken in the reaction. So this bond which is broken down, it is called as glycosidic bond. It is 1,4 glycosidic bond. 1,4 glycosidic bond means 1, 2, 3, 4. 1, 2, 3, 4. It is formed between carbon number 1 of one molecule and the carbon number 2 of the another molecule. So here you can see on this side there is carbon number 4 and on this side there is carbon number 1. So it is formed between 1,4 glycosidic bond, which is formed between one carbon number 1 of one molecule and the carbon number 4 of the other molecule. State the type of reaction catalyzed by maltase. So this is the hydrolysis. Because maltose has been broken down with the help of, in the presence of maltase by adding water into the 2 alpha glucose molecule. So this process is called as hydrolysis. Already told you the two types of reactions. The maltose was formed by the condensation reaction. Now, when we will add water into the maltose, it will be broken down into 2 glucose molecules. Let's move towards the next question. And next question is, in mammals, the small intestine is the main site of absorption of the products of digestion. Small intestine, you know, hai microvilli, where the absorption of the food takes place. So is a transmission electron micrograph of a longitudinal section of the part of an epithelial cell from the small intestine of a mammal. So figure 2.2. So they have given us the two pictures. Is a transmission electron micrograph of a horizontal section made at the position indicated by the two arrows in the figure 2.1. So here you can see the microvilli. The microvilli are in this way. Now I will move towards the question they have given us. So microvilli and cilia are cell structures. So the question is, describe how the structure of cilia differs from the structure of the microvilli visible in figure 2.1 and figure 2.2. In figure 2.1, microvilli, and in figure 2.2, they have shown us two pictures. So we have to differentiate between them. So describe how the structure of cilia differs from the structure of the microvilli visible in figure 2.1. So first of all, I would like to say that both cilia and microvilli are cell surface projections. There are fundamental differences in their internal structure, size, and function. So first of all, I will go with the cilia. So cilia are supported by a core of microtubules. Cilia are also very small in size. So cilia are supported by core of microtubules arranged in a characteristic 9+2 pattern. 9+2 pattern means 1, 2, 3, 4, 5, 6, 7, 8, 9. So in this way, So these are 9, 9 plus 2. So they will be arranged in this pattern, 9 plus 2 pattern. So these are the microtubules, which are 9 pairs of microtubules surrounding a central pair. So this is the central pair, and these are the 9 pairs which are around the central pair. So this complex is called an axonema. So in contrast, the microvilli are supported by a bundle of actin microfilaments that are not arranged in a specific patterns. You can see they are in this way. They are in this way. So if we talk about the differences between them, first of all, there is big difference in the structure. Difference in structure. Difference in structure. Secondly, their movement is also different. As you know, the microtubule structure and associated motor proteins, cilia are motile and can beat rhythmically. Cilia can beat rhythmically. So this movement is used to propel substance across the cell surface. So microvilli are non-motile extensions, whereas micro are non-motile. Secondly, if we talk about their size, cilia are typically longer and wider than microvilli. Longer than and wider. And function difference lead to different functions. The main purpose of is to increase the surface area. Microvilli are meant to increase the surface area. And for absorption. And cilia help in movement. And sense. They act as an antenna as well. So next question is, a scientist measured the length and the diameter of some of the microvilli shown in figure 2.1 to estimate the total surface area of microvilli on the surface of the epithelial cell. The scientist assumed the each microvillus was cylindrical in shape. This is just one other measurement needed to estimate the total surface area of the microvilli of the epithelial cell. So, well, uh, to estimate the total surface area of all the microvilli on a single epithelial cell, given the length and diameter of the individual microvilli, you need to know the total number of microvilli per epithelial cells. So, for this purpose, the scientist assumed each microvillus is a cylinder. Its surface area can be calculated by using its length and diameter. But the total surface area would then be the surface area of a single microvillus when we will multiply it with the total number of microvilli on the cell. So what we need to know the total number of microvilli. Given the length and diameter of individual and diameter is used to calculate the surface area of one microvilli. Which will be multiplied with total surface area. Which will be multiplied with total number of So in this way, you will be able to find out the total surface area for all microvilli. So it means we have to count total number of microvilli. Simply. Next question. Question number 2, part C. Identify the organelle labeled C in figure 2.1 and explain why there is a large number of these organelles in the epithelial cells of the small intestine, epithelial cells. The process of epithelial cells is so these are the organelles. So these are the organelles. These are the organelles, these. So these are these. So you can see their structure. So the structure shows that they are the mitochondria. Why mitochondria are here? Because the function of the epithelial cell is the absorption of the nutrients. So for this purpose, for this process of absorption, and the absorption takes place by the process of active transport. So organelle are mitochondria. So epithelial cells, are specialized for absorption of nutrients. So absorption of nutrients such as glucose and amino acids. Let me write. Very difficult to write, students on screen on the laptop. but I always try my best, but try to listen me as well. When I am explaining the questions, you can properly listen to me, then you will be able to understand. So epithelial cells are specialized for absorption of nutrients. Nutrients such as glucose and amino acid, which takes place by active transport. And you know, active transport is the movement of nutrients against the concentration gradient, that is from lower concentration towards the high concentration by using energy. So ATP is used, which is provided by mitochondria. So that is the reason many mitochondria are present in this area. Next question is, bacteria are found attached to epithelial cells in the intestine of mammals. Describe how the organization and distribution of DNA in epithelial cells differs from the organization and distribution of DNA in bacterial cells. Well, the organization and distribution of DNA in epithelial cells mean eukaryotes, and the DNA in the bacterial cells means prokaryotes. So we have to describe how they are different, or DNA organization and structure is different in both, that is in prokaryotes and in eukaryotes. So first of all, the location of these DNA molecules. Number one, as you know, in epithelial cells, DNA is located in nucleus. Whereas in bacteria, no nucleus, circular DNA is in cytoplasm. DNA is into multiple linear in epithelial cells. DNA is organized into multiple linear chromosomes. And tightly coiled around protein histone. And whereas in case of, the prokaryotes, the DNA is in the cytoplasm and you can say the single circular chromosome. But in bacteria, single circular chromosome without histone protein. So DNA is, in this case, DNA is not, and in bacteria, quantity large in eukaryotes or epithelial cells. Eukaryotes have more quantitatively as comparison to bacteria. So it means the both of they have the different location, they both of they have the different structure, and both of they have the quality wise different. One of them, eukaryotes or epithelial cells, they have histone proteins attached with them, whereas the other, they don't have the histone proteins attached with it. Quantity wise they are different, so in this way they are both of they are different. So it was all about the question number 1 and 2. Please don't forget to like, share, and subscribe my channel to watch more videos which are coming on the way. So please, thanks for watching.